Hello there, and welcome to Science for Sleep. Tonight, we'll explore the human brain. 86 billion neurons dancing across trillions of synapses, consciousness unfolding moment by moment. Every experience has shaped your mind, refined by millions of years of evolution into the most complex object in existence. You've got nothing to do but rest now. Let your mind relax, your breathing slow, your body grow light. There's nowhere to be, nothing to accomplish. Just you and the gentle rhythm of your own thoughts. If this helps you rest tonight, consider liking and subscribing to Science for Sleep. We're building a community of people who understand that rest is sacred. Your support helps us create more journeys into peaceful knowledge for everyone seeking gentle explorations of how our remarkable brains work. Now, let's begin our journey into the extraordinary landscape of the human brain. Within the soft hollow of your skull rests something so profoundly intricate that scientists continue to discover new mysteries about it even after decades of devoted study. Your brain contains approximately 86 billion neurons, a number so vast it mirrors the count of stars scattered across our entire Milky Way galaxy, like distant suns burning in the darkness. Each of these neurons does not exist in isolation, but rather reaches out delicately to thousands of others, creating connections called synapses that form an incomprehensibly complex web of communication. These trillions upon trillions of connections represent something almost sacred, a landscape of pure thought and possibility woven together by biology itself. Scientists sometimes call this architecture a neuron forest, an apt metaphor for the way these delicate branches intertwine and reach toward one another in the perpetual darkness of your skull. Imagine if you could somehow see inside this forest, you would witness an endless garden of electrical signals moving at remarkable speeds. The fastest of these signals travel at speeds approaching 250 miles per hour, faster than racing cars accelerating down infinite highways of biological circuitry. Yet despite this breathtaking speed, time still feels precious and unhurried when you are drifting off to sleep, as if your brain has learned to savor each passing moment. This vast network of neurons constitutes just one part of what makes your brain remarkable, for it is merely the foundation upon which all of your experience rests. As you settle deeper into relaxation now, know that these billions of messenger cells are already beginning to shift their patterns, preparing themselves for the profound rest that awaits you. During your lifetime, your brain performs something miraculous that was once thought to be impossible by neuroscientists who studied brain function with great care and attention. Your brain generates brand new neurons continuously, a process called neurogenesis that happens particularly in special regions like the hippocampus, where new memories first take root and begin to grow. This discovery transformed our understanding of the brain because, for many decades, researchers believed that you were born with a fixed number of neurons that only diminished with time and age. Yet emerging from mountains of research came this astonishing revelation that your brain remains capable of renewal, of creating fresh neural tissue even as you move through middle age and into your later years. These newly born neurons integrate themselves into existing networks, finding their place among their elder neighbors and contributing to ongoing processes of learning and memory formation. The hippocampus, that seahorse-shaped region nestled deep within your brain, seems to possess particular generative power, as if it holds the secret to perpetual youth and renewal. When you sleep tonight, these brand new neurons will be particularly active, consolidating their connections and settling into their roles within your vast neural landscape. This capacity for neurogenesis suggests something hopeful about aging and the human condition, that loss and renewal dance together in an endless waltz within your skull. Your brain's ability to birth new neurons means that you are never truly fixed in place, never entirely defined by your past, but always capable of becoming something slightly different and slightly new. This fundamental truth allows your mind to drift, now knowing that transformation and growth continue even in silence and in sleep. The electrical signals that traverse your brain's pathways do not travel in simple straight lines, but rather follow complex routes shaped by the architecture of your neural networks with remarkable precision. These signals move along structures called axons, which are thin projections extending from neurons that can be as long as your arm in some cases, carrying messages across vast distances within the brain and body. When these electrical impulses reach the end of an axon, they encounter a narrow gap called a synapse, where the signal must somehow cross to reach the next waiting neuron. This crossing happens through a dance of chemistry and electricity as delicate as the unfurling of a flower petal in spring. Neurotransmitters, which are chemical messengers released from the sending neuron, drift across the synaptic gap and attach themselves to receptors on the receiving neuron like keys sliding into locks. If the signal is strong enough and the receiving neuron is sufficiently primed, it will fire its own electrical impulse, continuing the conversation between brain cells. This process happens billions of times every second throughout your waking and sleeping hours, creating the substrate of all thought and sensation. The remarkable thing about synapses is that they are not rigid and unchanging, but rather flexible and dynamic, capable of strengthening or weakening based on patterns of activity. When synapses strengthen through repeated activation, a process begins that underlies learning and memory formation, as if your experiences were literally reshaping your brain structure. As your brain settles towards sleep, this synaptic activity will begin to shift and transform, preparing your neurons for the restorative work of rest and consolidation. Within the cellular fabric of your brain lies a remarkable structure that was once thought too rare and too isolated to matter significantly in brain function, yet scientists now recognize it as something extraordinary. The Purkinje cell, found predominantly in a wrinkled region called the cerebellum, possesses perhaps the most extensively branched dendritic tree of any neuron in the entire human brain. A single Purkinje cell can form connections with up to 200,000 other neurons, an astonishing degree of connectivity that allows it to integrate vast amounts of information and coordinate complex patterns of activity. These cells sit in the cerebellum like master orchestrators, fine-tuning your movements, your balance, and your coordination with exquisite precision, even when you are not consciously aware of their work. When you sleep, these Purkinje cells remain partially active, their vast networks humming quietly in the background as your brain processes the day's experiences. The architecture of the Purkinje cell serves as a reminder that brain complexity exists at multiple levels, from the microscopic dance of synaptic transmission to the grand orchestration of movement and sensation across whole systems. Your cerebellum, though smaller than the cerebrum above it, contains roughly as many neurons as the rest of your brain combined, a hidden universe of neuronal tissue devoted to the smooth coordination of all your physical experiences. As you rest now and allow your body to relax, the Purkinje cells within your cerebellum are already easing their activity, releasing their vigilant grip on muscular control. This gentle loosening of cerebellar activity marks one of the first steps toward true sleep, a softening of the brain's constant surveillance over bodily movement and position. The substance of your brain itself is composed largely of something that might surprise you, given what you have learned about neurons and electrical signals and synaptic transmission. Your brain is approximately 60% fat, making it the fattiest organ residing anywhere within your entire body, a remarkable fact that seems to contradict the popular image of the brain as a purely electrical and chemical entity. This fat is not inert or merely structural, but rather absolutely essential for the functioning of your neural tissue because it insulates the axons of neurons in a protective sheath called myelin. This myelin covering works much like the insulation around an electrical wire, preventing signal leakage and allowing neural impulses to travel faster and with greater efficiency across long distances. Without adequate fat, and particularly without the specialized fats called lipids and cholesterol that your brain manufactures within its own tissues, your neurons would function sluggishly and your thoughts would move with great difficulty. The brain is so dependent on cholesterol that approximately 25% of all the cholesterol in your entire body resides within this 3-pound universe contained in your skull. When myelin breaks down with age or injury, your brain's signaling capacity diminishes and cognitive problems can emerge, which is why maintaining healthy fat metabolism appears crucial for preserving brain function across the lifespan. This reliance on fats and cholesterol means that the brain's health is intimately connected to your overall nutritional state and metabolic wellness. As you settle into sleep tonight, know that this fatty tissue surrounding your neurons is serving you even now, enabling the smooth flow of electrical signals that will process and consolidate your day's experiences into memory. Your brain's fat is not something to fear, but rather something to honor, as it is the very medium through which your thoughts take flight. The journey from infancy to full adult brain maturity spans a surprisingly long period of your life, extending well into your 20s and reminding us that human development is a gradual and patient process. When you were born, your brain was only a quarter of its adult weight, a soft and relatively undeveloped organ with vast potential still unrealized within its neural tissues. Within just the first year of life, your brain roughly tripled in size as billions of new connections formed between neurons at a breathtaking pace. Each experience, you had literally building the physical structure of your mind. By the time you reached age 2, your brain had already attained approximately 80% of its adult size, though its internal organization and complexity continued developing for years to come. The different regions of your brain matured at different rates, with some areas reaching their final form in childhood, while others continued developing through adolescence and into early adulthood. The prefrontal cortex, that region responsible for decision-making, impulse control, and abstract reasoning, was among the last to fully mature, not reaching its complete development until approximately age 25. This extended maturation period meant that teenagers had brains that were still under construction, particularly in the areas governing judgment and self-control, which explains many aspects of adolescent behavior that puzzled generations of parents. As you reach your current age, the major structural development of your brain has largely stabilized, though your brain continues to reshape and reorganize itself throughout life through the processes of learning and experience. This long developmental timeline reminds us that human beings are creatures of gradual unfolding, not sudden transformation, and that patience with ourselves and others honors the biological truth of human development. Tonight, as you sleep, your brain will continue this quiet work of maintenance and gradual refinement, tending to its networks with the patience that marks all truly deep processes. The act of remembering is not a simple retrieval process like pulling a file from a cabinet, but rather a complex and remarkable symphony of neural activity that your brain conducts with extraordinary grace. When you form a memory, particularly in the hippocampus, that seahorse-shaped region deep within your brain does something profoundly elegant called binding. It takes separate features of an experience, the sights and sounds and emotions and physical sensations that occurred together, and weaves them together into a unified whole. This binding process allows you to later relive an experience not as isolated fragments, but as an integrated moment that feels continuous and coherent. When you remember a friend's face, you don't retrieve just their facial features, but rather, your brain reconstructs the entire context of knowing them, your emotions upon seeing them, and the experiences you shared together. The hippocampus appears to serve as a temporary binding site where these separate information streams converge and are woven together into episodic memories that can later be stored more permanently in the cortex. This binding happens through patterns of neural activity called neural ensembles, where groups of neurons fire together in specific synchronized patterns that encode the essence of the moment. Your brain's remarkable ability to bind disparate elements into cohesive memories might explain why dreams during sleep can feel so vivid and emotionally intense, as your brain practices its binding and integration processes in the absence of external input. As you approach sleep, your hippocampus will gradually shift into a different mode of activity, preparing to consolidate the day's experiences into long-term storage. This shift marks a gentle transition from the gathering of experience to the integration and storage of what has been gathered, a movement from outward attention to inward reflection and organization. The connections between neurons are not static structures forged once and then remaining unchanged forever, but rather dynamic living structures capable of strengthening or weakening based on patterns of neural activity. This property is called synaptic plasticity, and it represents one of the most fundamental principles underlying how your brain learns and adapts to new experiences. When synapses are repeatedly activated through patterns of neural firing, they tend to strengthen, becoming more sensitive and responsive to future signals. Much like a well-worn path through a forest becomes easier to traverse. This strengthening process is sometimes summarized with the phrase neurons that fire together, wire together, capturing the essence of how learning physically reshapes brain structure at the synaptic level. Conversely, synapses that remain unused tend to weaken and fade, a process of pruning that helps your brain eliminate unnecessary connections and maintain efficiency. This constant remodeling of synaptic strength means that your brain is literally being reshaped by everything you experience and everything you practice and everything you attend to with focused attention. Synaptic plasticity underlies all forms of learning, from mastering a new skill to understanding a new concept to recovering function after brain injury. The brain's flexibility in this regard offers hope to people dealing with neurological challenges because damaged circuits can potentially be rewired and compensated for through dedicated practice and experience. As you sleep tonight, many of your synapses will undergo subtle changes as your brain consolidates memories and integrates new learning into your existing knowledge base. The very structure of your brain will be different tomorrow morning than it was today, altered by every experience and every moment of rest and every thought that flickered through your consciousness. During the night when you surrender to sleep, your brain experiences something that many people would not expect, given that sleep feels like a time of rest and dormancy. Your brain enters periods called REM sleep, where its activity levels rise to heights that nearly rival your waking consciousness, as if your brain becomes dramatically more active precisely when your body is most still. REM stands for Rapid Eye Movement Sleep and refers to the distinctive pattern of eye movements that occur beneath closed eyelids during these vivid, dream-filled portions of your sleep cycles. During REM sleep, your brain floods itself with a neurotransmitter called acetylcholine, while simultaneously reducing the levels of serotonin, norepinephrine, and histamine in your neural tissues. This precise chemical shift creates the conditions for vivid dreaming because the reduced serotonin and norepinephrine mean that the experiences generated by your dreaming brain do not transfer into permanent memory in the way that waking experiences do. Yet this is not a flaw, but rather a feature of your brain's design, as it allows you to explore and process experiences in the dream world without the waking world becoming cluttered with false memories. Your eyes move rapidly during REM sleep, darting back and forth as if you were watching the events unfolding in your dreams, yet your large muscles remain in a state of temporary paralysis. This paralysis seems to be a protective mechanism preventing you from acting out your dreams and potentially harming yourself or others. As you approach true sleep tonight, your brain will cycle through several REM periods, each one becoming longer and more prominent as the night progresses. In these REM stages, your brain will be remarkably active, processing emotions, consolidating learning, and weaving the threads of your day's experiences into the fabric of your long-term memory. The organization of your brain into separate hemispheres, each with somewhat specialized functions, creates a fascinating duality within your skull that allows for remarkable flexibility and parallel processing. Your left and right cerebral hemispheres are connected by a massive bundle of nerve fibers called the corpus callosum that contains approximately 200 million axons, creating a communication superhighway between the two halves. Though the hemispheres are roughly mirror images in appearance, they are functionally asymmetrical in ways that remain partly mysterious even to modern neuroscience, with some functions clearly associated with one side while others remain distributed. The left hemisphere, in most people, contains specialized areas devoted to language production and comprehension, as well as logical reasoning and sequential processing, allowing your brain to understand words and construct coherent thoughts. The right hemisphere, conversely, appears specialized for visual spatial abilities, abstract reasoning pattern recognition, and the integration of emotional information, allowing your brain to grasp the bigger picture and perceive relationships that extend beyond simple logical analysis. This functional specialization does not mean that the hemispheres work independently, for they are in constant communication through the corpus callosum, sharing information and coordinating their activities in ways that produce unified consciousness. When one side of your brain sustains injury, the other side sometimes demonstrates a remarkable capacity for compensation, gradually taking on functions that were previously handled by the damaged hemisphere. This hemispheric complementarity suggests that your brain is organized for both specialization and flexibility, capable of deep expertise in particular domains while also maintaining the capacity to adapt and reorganize when circumstances demand it. As your brain settles into sleep, this hemispheric dance continues unabated, with each hemisphere contributing its unique perspective to the processing and consolidation of your day's experiences into enduring memories. Your brain's weight and proportions relative to your body size place you among the most cognitively endowed creatures to have ever existed on Earth, a distinction that connects you to millions of years of human evolution. Your adult brain weighs approximately 3 pounds, only 2% of your total body weight, yet it consumes approximately 20% of your body's total energy expenditure. This extraordinary metabolic demand reflects the immense activity constantly occurring within your neural tissue as billions of neurons fire and billions of synapses transmit signals throughout the day and night. The human brain is roughly 3 times larger in overall volume than the brains of our closest living relatives, the chimpanzees and gorillas. And this size difference corresponds to significant increases in cognitive capacity, particularly in areas related to language reasoning and abstract thought. Evolution has invested enormous resources in building and maintaining this large, expensive brain, a biological commitment that speaks to the tremendous advantages that advanced cognition provides to the human species. The size of your brain relative to your body represents the culmination of millions of years of natural selection, favoring larger brains with more sophisticated organization and more extensive connections. This remarkable evolutionary achievement means that the 3-pound organ within your skull contains not just neurons and synapses, but also millions of years of evolutionary wisdom encoded in its structure and function. As you drift towards sleep tonight, you can take comfort in knowing that this brain of yours represents the height of what evolution has been able to produce on this planet, a culmination of deep time compressed into the soft tissues of your skull. This knowledge can invite a kind of reverence and respect for the organ that has carried you through all of your experiences and that continues to think and feel and dream even as you rest. Deep within the delicate tissues of your brain lies a complexity that continues to astonish neuroscientists who dedicate their entire careers to understanding how neurons communicate and organize themselves. The brain's synapses, those tiny spaces where one neuron speaks to another, contain over 1,800 different types of proteins working in concert to enable neural signaling. Each protein serves a unique and specialized purpose, from anchoring neurotransmitter receptors to recycling used neurotransmitters back into the synapse to strengthening or weakening connections based on patterns of activity. This astounding diversity of proteins means that synapses are not simple uniform structures, but rather incredibly sophisticated machines, each one finely tuned by evolution to perform its specific role within the larger neural network. When you consider that your brain contains trillions of synapses, and each synapse contains thousands of proteins of dozens of different types, the sheer computational complexity becomes almost incomprehensible. It reminds us that the brain's sophistication operates not just at the level of individual neurons or large brain regions, but also at the molecular and protein level, where countless intricate interactions orchestrate the work of thought and sensation. This molecular complexity underlies every experience you have ever had and every memory you have ever formed. As you drift deeper into relaxation now, know that this molecular symphony continues even as your conscious mind settles and quiets, these proteins working tirelessly to maintain and reorganize the structure of your thoughts. When darkness falls and you finally surrender to sleep, something remarkable happens within your brain that might seem counterintuitive, given that sleep feels like a time of rest and mental quietude. Your brain initiates a cleaning process called the glymphatic system that operates most efficiently during the deepest stages of sleep, when your brain can finally pause its constant vigilance. This system works like an internal janitorial service, clearing away toxic waste products and metabolic byproducts that accumulate throughout your day as billions of neurons fire and consume energy. The cerebrospinal fluid that bathes your brain increases its circulation during sleep, flushing through neural tissue and literally washing away proteins like amyloid beta that, if allowed to accumulate, could contribute to neurodegenerative diseases. Remarkably, your brain cells actually shrink by about 60% during deep sleep, creating larger spaces between them through which cerebrospinal fluid can flow more freely and efficiently. This shrinking of brain cells during sleep seems to be an active process coordinated by your brain's glial cells, which support and regulate neuron function. Without this nightly cleaning process, harmful proteins would gradually build up and interfere with neural signaling and cognitive function over time. The glymphatic system represents one of the most important discoveries about sleep in recent decades because it explains at least part of why sleep deprivation has such profound negative effects on brain function and cognitive ability. As you settle into sleep tonight, this glymphatic system will activate and begin its crucial work of maintaining your brain's long-term health. This knowledge can help you approach sleep not as a luxury, but as a biological necessity, as essential to your health as eating or breathing. The energy demands of your brain are so extraordinary and unrelenting that they shape a significant portion of your body's overall metabolism and nutritional requirements. Your brain consumes approximately 20% of your body's total energy expenditure, despite representing only about 2% of your body's total weight, a metabolic demand that reflects the incredible activity constantly occurring within its neural tissues. This energy consumption supports the constant firing of neurons, the maintenance of ion gradients across neuronal membranes, the synthesis of neurotransmitters, and the consolidation of memories, among countless other processes. A single neuron can fire hundreds of times per second, and with 86 billion neurons, many of them firing simultaneously, the total energy demand becomes staggering. If your brain were not able to access a constant supply of glucose and oxygen, it would begin to fail within minutes, much faster than other organs like your heart or muscles. This vulnerability to energy deprivation explains why unconsciousness occurs so rapidly when blood flow to the brain is interrupted, and why brain damage from oxygen deprivation can be so severe. It also explains why sleep is so important for brain health, because during sleep, energy consumption decreases, and the brain can engage in restorative processes like the glymphatic cleaning without the constant demands of maintaining consciousness and processing sensory information. As you move deeper into sleep tonight, your brain's energy consumption will gradually decrease, allowing some of that energy to be redirected toward repair and restoration rather than the constant work of waking consciousness. This shift in energy allocation marks one of the most important transitions between wakefulness and sleep. The rhythm of a REM sleep follows a pattern throughout the night that reveals something profound about how your brain organizes sleep and prepares memories for long-term storage. Your first REM sleep period occurs approximately 60 to 90 minutes after you fall asleep and lasts only about 10 minutes, a brief window into the dream world that feels almost like an appetizer before the main course. As the night progresses and you cycle through repeated stages of sleep, your REM periods become progressively longer and more vivid, as if your brain is gradually settling into deeper and more intense dream work. By the end of a typical 8-hour sleep period, your final REM cycles can last an hour or more, providing an extended opportunity for your brain to process and consolidate the day's complex emotional and cognitive experiences. This lengthening of REM periods through the night suggests a deliberate strategy on your brain's part to devote more resources to dream-based processing as the night advances. The brain appears to prioritize the restoration and consolidation of emotional memories and complex learning during these final extended REM periods when external distractions and the demands of waking life have receded. Understanding this pattern can help you appreciate that if you cut your sleep short, you sacrifice the longest and potentially most restorative REM periods, which occur only toward the end of the night. As you begin your sleep tonight, your brain will follow this ancient pattern, beginning with brief REM exploration and gradually extending these periods as the night deepens. This progressive lengthening of REM sleep represents one of your brain's most sophisticated adaptive strategies for managing the vast volume of information and emotion that accumulates throughout your waking day. The thalamus is a relatively small paired structure buried deep within your brain, yet it performs a role so central to your consciousness that damage to it can result in profound alterations to your awareness. This structure serves as a relay station and integration hub where information from your senses converges and is coordinated before being sent to the cerebral cortex for processing and conscious perception. Nearly all sensory information from your eyes, ears, and skin passes through the thalamus, which acts as a sophisticated filter and organizer, deciding which information deserves to be processed further and which can be ignored. The thalamus also plays a crucial role in regulating consciousness itself, participating in the wake-sleep cycle and helping to determine whether you remain conscious or slip into deeper sleep. Remarkably, the thalamus contains about 50 million neurons, which, though substantial, represents only a tiny fraction of your brain's total neuron count, yet its influence on your consciousness is profound and far-reaching. During REM sleep, the thalamus shows a distinctive pattern of activity that appears to facilitate the vivid hallucinations and dream experiences you have during this stage of sleep. The thalamus seems to maintain consciousness and coherence by binding together separate streams of sensory information into a unified conscious experience that feels continuous, even though it actually consists of many separate neural processes. As your brain moves through different sleep stages tonight, the thalamus will shift its activity patterns, helping to coordinate the transition from the processing demands of wakefulness to the restorative work of deep sleep. Understanding the thalamus's role in consciousness can deepen your appreciation for the remarkable feat of neural integration that allows you to experience the world as a unified whole rather than as fragmented and disconnected sensations. The remarkable ability of your brain to reshape itself in response to experience and learning is called neuroplasticity, and it represents one of the most hopeful discoveries in modern neuroscience. Neuroplasticity means that the physical structure of your brain today is different from the physical structure it had a year ago because every experience you have, every skill you practice, and every thought you think literally rewires your neural circuits. When you learn something new, whether it is a language, a musical instrument, or a physical skill, your brain forms new connections between neurons, strengthens existing synapses, and sometimes even reorganizes which regions handle particular functions. This reorganization can be quite dramatic, as demonstrated by studies of musicians whose auditory cortex is physically enlarged compared to non-musicians or people who learn to read late in life and show distinctive patterns of neural reorganization. Neuroplasticity also provides hope for recovery after brain injury because damaged neural circuits can sometimes be compensated for through the formation of new pathways and the recruitment of other brain regions to handle functions previously localized elsewhere. The brain remains plastic throughout your entire life, though the degree and ease of neuroplasticity does gradually decrease with age as your neural networks become more established and less flexible. Yet even in very old age, the brain retains the capacity to learn and reorganize itself, suggesting that aging need not mean stagnation, but rather a shift toward different modes of growth and adaptation. As you sleep tonight, your brain will engage in neuroplastic processes, consolidating new learning and reorganizing neural circuits based on your day's experiences. The very act of sleep itself appears to be a crucial mechanism through which your brain implements the changes that allow neuroplasticity to occur. The structure of individual synapses can change their strength and responsiveness through a process called synaptic plasticity that operates across timescales ranging from milliseconds to hours to days. Long-term potentiation is one of the most well-studied forms of synaptic plasticity, and it refers to a persistent strengthening of a synapse that occurs when neurons are repeatedly activated in a particular pattern. When two neurons fire together repeatedly, the connection between them tends to strengthen such that the presynaptic neuron becomes increasingly effective at driving the postsynaptic neuron to fire as well. This strengthening appears to occur through multiple mechanisms, including the insertion of additional neurotransmitter receptors on the postsynaptic membrane and the increased release of neurotransmitters from the presynaptic terminal. Long-term potentiation is thought to be a fundamental mechanism underlying learning and memory because it literally encodes experience into the structure of your neural circuits. Conversely, long-term depression refers to a weakening of synaptic connections that occurs when neurons fire in particular patterns or when there is a mismatch between expected and actual neural activity. The balance between strengthening and weakening of synapses is crucial for proper brain function because excessive strengthening could lead to rigid, inflexible thinking, while excessive weakening could prevent the formation of memories. As you learn throughout your day, synaptic potentiation is constantly occurring, creating physical traces of your experiences within your neural tissue. During sleep, this synaptic plasticity continues in a more deliberate and organized fashion as your brain consolidates the day's learning and integrates new information into existing knowledge structures. The delicate finger-like projections that emerge from neurons called dendritic spines represent the actual sites where most synaptic connections occur, and changes in dendritic spine morphology appear closely linked to memory formation and cognitive function. These spines are incredibly dynamic structures that can extend, retract, enlarge, and shrink over time scales ranging from minutes to hours, reflecting the constant reorganization of neural circuits. There are thin spines and mushroom-shaped spines, with the mushroom spines tending to be larger, more stable, and more functionally important for transmitting signals, while thin spines are more variable and appear to represent connections that are being actively modified. Studies show that when animals learn something new, the density and morphology of dendritic spines changes in brain regions related to what was learned, and this structural change correlates with how well the new information was retained. With aging, dendritic spines tend to become less stable, particularly thin spines, which may explain some of the cognitive slowing that occurs in older adults, though this remains an area of active research. Interestingly, mushroom-shaped spines appear somewhat resistant to age-related changes, suggesting that the brain's capacity to maintain well-established, important connections remains relatively preserved, even as the ability to form new connections gradually declines. The remarkable thing about dendritic spines is that they reveal learning and memory as fundamentally physical processes, not abstract or merely chemical, but literally involving the sculpting of your brain's physical architecture. As you rest tonight, new dendritic spines are forming in your brain even during sleep, particularly in regions where you learned something new during the day. Deep sleep, which is called Stage 3 or N3 sleep, represents the most restorative phase of your sleep cycle when your brain engages in its most intensive repair and restoration work. During this stage, your brain waves become slow and powerful, with a distinctive pattern called delta waves that have a frequency of only 1 to 4 hertz compared to the much faster rhythms of waking consciousness. The slower brain waves of deep sleep appear to reflect a shift away from the constant scanning and processing of information that characterizes waking and toward an inward-focused state of consolidation and restoration. Your body temperature drops significantly during deep sleep. Your muscles relax more deeply than at any other time, and your heart rate and breathing slow to their lowest levels of the night. Growth hormone is released during deep sleep at much higher rates than during waking hours, and this hormone drives physical restoration, muscle protein synthesis, and other restorative processes throughout your body. Your immune system is also particularly active during deep sleep, with immune cells patrolling your body and fighting off infections more effectively. Despite the low level of overall brain activity during deep sleep, this stage is crucial for memory consolidation, creativity, and problem solving as your brain processes and integrates the information gathered during waking hours. The amount of deep sleep you obtain each night appears strongly linked to whether you wake up feeling truly rested and refreshed, with insufficient deep sleep leaving you feeling tired and drained even if you spent 8 hours in bed. As you approach the earlier stages of sleep tonight, your brain will gradually deepen until it reaches the profound restoration of Stage 3 sleep. This shift represents a sacred transition from the external world of waking awareness to the internal world of neural restoration and repair. The communication between your left and right cerebral hemispheres occurs through a massive structure called the corpus callosum that contains roughly 200 million axons functioning as an information superhighway. This incredibly dense bundle of nerve fibers allows the two hemispheres, which are remarkably specialized and distinct in their functions, to coordinate their activities and share information about what each is processing. The corpus callosum appears crucial for integrating information from the two hemispheres into a unified consciousness, so that even though your brain is divided into left and right halves, your conscious experience feels continuous and unified. Damage to the corpus callosum disrupts the flow of information between hemispheres and can result in fascinating neurological phenomena where the left and right hemispheres seem to operate almost independently. Studies of patients with severed corpus callosa have revealed that consciousness itself may not be as unified and singular as we normally assume, but rather represents an integration of multiple parallel processing streams. The development of the corpus callosum in infants appears tied to the development of coordinated body movements and integrated perceptual experiences, suggesting that this structure enables the remarkable coordination between the two sides of your body and brain. It reminds us that the brain's sophistication depends not just on what individual regions can do, but also on how effectively different regions communicate and integrate their information. During sleep, the corpus callosum remains active, maintaining coordination between hemispheres even as the overall level of consciousness diminishes. As you drift toward sleep, your brain's hemispheres will continue their constant dialogue through this remarkable structure, maintaining the unified sense of self that persists even in dreams. Your brain's growth and development follow a precisely orchestrated schedule that extends from before birth through early adulthood, and these developmental trajectories shape your cognitive abilities and personality in profound ways. When you were born, your brain was remarkably immature, containing most of the neurons you would ever have, but with very few of the connections between them established. Within just the first year of your life, your brain approximately tripled in weight as connections between neurons multiplied at an astonishing rate in response to your experiences, interactions, and sensory stimulation. This period of explosive connection formation, called synaptogenesis, represents the most intense period of brain development you will ever experience, with peak synaptic density reached around age 3. Over childhood, your brain underwent a process of selective pruning, where connections that were rarely used were eliminated, while connections that were frequently activated were strengthened and stabilized. Different brain regions matured at different rates, with primary sensory regions becoming relatively mature by early childhood, while association areas like the prefrontal cortex remained under construction well into adolescence. The maturation of the prefrontal cortex, which governs decision-making, planning, and impulse control, appeared to be one of the last major developmental processes, extending into the mid-20s. This extended development period meant that adolescents had brains literally under construction, particularly in regions responsible for judgment and self-control, which helps explain many aspects of teenage behavior that had puzzled adults. As you sleep tonight, you are not simply resting, but engaging in processes that originated during infancy and continue to shape your brain throughout your entire life. This continuity connects your sleep tonight to the sleep of the infant you once were, and to the ongoing development that still shapes who you are becoming. As you settle deeper into the early stages of relaxation, your awareness beginning to soften and distance itself from the concerns of the day, consider the remarkable journey that genes within your brain have taken across millions of years of evolution. Some genes expressed in the human brain show patterns and expression levels completely unique to humans never observed, with exactly the same characteristics in the brains of other primates or animals. These uniquely human genes often relate to functions that are distinctly human, like language, abstract reasoning, extended childhood development, and sophisticated social cognition. Many of these distinctly human genes are involved in building and maintaining synapses, the connections between neurons, suggesting that the way our brains wire themselves together is fundamentally different from how other brains organize their neural networks. Some of these unique genes are expressed not in the neurons themselves, but rather in support cells called astrocytes and microglia that help nurture and maintain neurons. This tells us that human consciousness and cognitive sophistication arise not just from what individual neurons do, but from how the entire cellular ecosystem of the brain is organized and coordinated. These genetic differences between human brains and other brains remind us that we are the products of evolution, refined over millions of years into something genuinely novel and unprecedented. As your mind drifts deeper now, you can take comfort in knowing that the unique genetic wisdom encoded within your brain represents millions of years of evolutionary refinement. The most abundant neurotransmitter in your brain and nervous system is called dopamine, and it plays roles so diverse and important that disruptions in dopamine signaling underlie some of the most serious neurological and psychiatric conditions. Dopamine is involved in the brain's reward system, creating the pleasurable sensations you experience when you achieve something meaningful or when good things happen in your life. Beyond pleasure, dopamine plays crucial roles in movement control, with certain neurons using dopamine to regulate your body's physical actions and coordination. Dopamine is also essential for motivation and goal-directed behavior. Without adequate dopamine, people struggle to initiate action or persist toward long-term goals, and life becomes a gray landscape lacking drive and meaning. Your brain produces dopamine in specific regions like the substantia nigra and ventral tegmental area, which then send out projection fibers that spread throughout the brain like roots of a tree reaching toward light. When dopamine production or signaling becomes disrupted, as occurs in Parkinson's disease, the tremors and movement difficulties reflect the disruption of these dopaminergic pathways. Depression and many mood disorders appear linked to dysregulation of dopamine systems, and medications that affect dopamine, such as stimulants, can have profound effects on mood motivation and behavior. Even now, as you rest, dopamine continues to circulate through your brain, supporting your sense of well-being and the quiet satisfaction of rest. As you move deeper toward sleep, the dopamine system will gradually quiet, allowing other neurotransmitter systems to take the lead in orchestrating your brain's transition to sleep. Another neurotransmitter of profound importance, called serotonin, influences so many aspects of your mental and physical life that it is sometimes called the brain's mood molecule. Though this nickname oversimplifies its truly vast sphere of influence. Serotonin is produced primarily in small clusters of neurons in the brainstem called the raphe nuclei, from which it projects throughout the cerebral cortex, the limbic system, and many other brain regions. Serotonin influences your mood, affecting whether you feel calm and content or anxious and dysphoric, and dysregulation of serotonin is implicated in depression, anxiety, and obsessive-compulsive disorder. Beyond mood, serotonin regulates your sleep-wake cycles, helping to regulate circadian rhythms and promoting the transition into sleep when serotonin levels rise in response to darkness. Serotonin also influences appetite and in the hypothalamus region of your brain. It helps regulate how hungry or full you feel. The neurotransmitter affects cognitive functions, including memory formation and learning and dysfunction. In serotonergic systems appears linked to cognitive decline in aging. SSRIs Selective serotonin reuptake inhibitors are medications that increase serotonin levels in the brain by blocking the reuptake of serotonin back into presynaptic terminals, allowing serotonin to act longer in the synaptic space. As you approach sleep tonight, serotonin is already beginning to rise in your brain, promoting the sense of calm and the biological shift from wakefulness toward slumber. The seahorse-shaped structure called the hippocampus is essential for the formation of new memories, particularly declarative memories, which are memories of facts and events that you can consciously recall and deliberately retrieve. When you learn new information, whether historical facts, scientific concepts, or personal experiences, the hippocampus plays a central role in binding the disparate elements of that information into a coherent memory that can later be stored and retrieved. The hippocampus also associates spatial information with memories, meaning that many of your memories include a sense of where they occurred and the spatial context in which they unfolded. Damage to the hippocampus, as occurred famously in a patient called H.M., who had his hippocampus surgically removed, results in profound difficulty forming new memories, while older memories stored in the cortex remain largely intact. Remarkably, the hippocampus seems less important for maintaining memories long-term once memories have been consolidated in the cortex than it is for the initial formation process. The hippocampus continues to be involved in memory retrieval, suggesting that it serves as a kind of index or catalog that helps retrieve stored memories from across the cortex. This remarkable structure sits at a crossroads where information from multiple brain regions converges, allowing it to integrate diverse aspects of experience into unified memories. As you sleep tonight, your hippocampus will be particularly active, consolidating the day's experiences into memories that will fade by morning or persist into the weeks and years ahead, depending on their significance. When you drift into stage 3, sleep called deep sleep or slow wave sleep, your brain enters a state of reduced consciousness and profound restoration that feels almost like a little death, from which you periodically resurrect. During deep sleep, your brain waves become exceptionally slow, showing a pattern called delta waves that have a frequency of only 1 to 4 cycles per second compared to the 13 to 30 cycles per second you experience during waking. These slow delta waves appear to reflect synchronized activity across large populations of neurons firing together in coordinated patterns quite different from the desynchronized activity of waking consciousness. Deep sleep is the most restorative sleep stage where your body engages most intensively in physical repair and recovery, with growth hormone secretion at its peak. Your immune system is also highly active during deep sleep, with immune cells patrolling your body and mounting defenses against pathogens more effectively than during waking hours. Memory consolidation occurs during deep sleep, particularly the consolidation of declarative memories and procedural memories, through mechanisms that are still being actively researched. This stage of sleep is crucial for feeling truly rested, and the more deep sleep you obtain each night, the more likely you are to wake feeling fully refreshed and restored. As you approach the deeper stages of sleep tonight, you will experience this profound shift toward restoration as your brain slows and your consciousness recedes into the depths of this most restorative stage. The striatum is a structure deep within your brain that receives projections from dopamine neurons and plays a crucial role in movement control, reward processing, and habit formation. When dopamine is released into the striatum, it affects how strongly neurons fire and how readily movements are initiated, and this dopaminergic signaling is essential for normal movement. The striatum is also part of your brain's reward and motivation system, with dopamine release in the striatum, creating the feeling of pleasure or satisfaction that reinforces behaviors that led to positive outcomes. The striatum also appears involved in learning habits and routines, so that actions you perform repeatedly gradually shift from requiring conscious attention to unfolding automatically. Disruptions in striatal dopamine signaling, as occurs in Parkinson's disease, result in difficulty initiating movement, causing the characteristic tremors and rigidity that condition. The striatum also receives input from the prefrontal cortex, which helps to regulate goal-directed behavior, ensuring that your actions remain aligned with your intentions and values. This subcortical structure represents a crucial hub in your brain's control of movement, motivation, and learning. As you relax now, the striatum will gradually quiet its signaling, allowing your body to release its muscular tension and prepare for the paralysis of REM sleep. Consciousness itself is one of the deepest mysteries in neuroscience, and some scientists believe that consciousness arises from coordinated activity between the thalamus and cortex working together to bind separate streams of information into unified awareness. The hippocampus also appears crucial for conscious experience, particularly for the conscious recollection of episodic memories and the sense of personal continuity across time. Memory and consciousness are intimately intertwined, with the systems that create and maintain memories also being involved in generating and maintaining conscious experience. When the thalamus fails to properly relay and integrate information, consciousness is disrupted, and people may fall into unconscious states or experience disorders of consciousness. The binding of information from diverse sources into a unified conscious experience appears to be something your brain actively constructs moment by moment, rather than something that passively emerges from the physical structure. This binding or integration function requires communication between multiple brain regions, particularly between thalamic relay stations and cortical processing areas. The wonder of consciousness arising from mere electrochemical activity of billions of neurons connected by trillions of synapses represents one of the greatest mysteries remaining in science. As you drift now towards sleep, you will gradually experience a dissolution of consciousness as the thalamic cortical systems that generate waking consciousness shift into the modes characteristic of sleep and dreaming. In the first few stages of sleep, called Stage 1 and Stage 2, light sleep, your brain gradually transitions from the processing of external information toward inward-focused activity, while your conscious awareness of the outside world fades. Stage 1. Sleep typically lasts only a few minutes and represents a drowsy state in which you are just beginning to disengage from wakefulness. Your muscles still retain some tone, and you can be easily awakened. During Stage 1, your brain waves begin to slow, showing theta wave activity that marks the transition from waking alertness towards sleep, with your eyes moving slowly beneath your eyelids. Stage 2. Sleep lasts longer than Stage 1, and your brain becomes progressively less responsive to external stimuli, though you can still be awakened with effort, and your muscles continue to gradually relax. During Stage 2, your brain waves continue to slow, but are interspersed with brief bursts of faster activity called sleep spindles that appear important for memory consolidation. Your body temperature begins to drop during light sleep, and your heart rate and breathing gradually slow as your autonomic nervous system shifts toward parasympathetic dominance. These light sleep stages serve as a bridge between wakefulness and the deeper, restorative stages of sleep, allowing your brain to gradually transition rather than abruptly switching off. As you begin your sleep tonight, you will pass through these gentle, light sleep stages as your brain gradually shifts its gears and your conscious connection to the outside world softly fades. The cerebellum is a region at the back of your brain that is remarkably densely packed with neurons and plays essential roles in coordinating movement, balance, and even in some types of learning and planning. The cerebellum contains particular cell types, like the famous Purkinje cells, whose dense connectivity allows them to integrate vast amounts of information and precisely orchestrate motor commands. When you perform any coordinated movement, whether walking, running, catching a ball, or typing, the cerebellum is continuously monitoring and refining that movement, comparing your actual performance to your intended movement, and making tiny adjustments. The cerebellum receives input from your sensory systems, providing information about where your body is in space and how your muscles are contracting, and sends output back to motor regions that control your muscles. Damage to the cerebellum results in ataxia, a condition characterized by uncoordinated clumsy movements, loss of balance, and difficulty with precise motor tasks. The cerebellum has also been implicated in some cognitive and emotional functions, suggesting that its role extends beyond simple motor coordination to higher-level functions. During sleep, the cerebellum remains partially active, continuing its work of maintaining coordination, even though your muscles are largely paralyzed during REM sleep. As your body relaxes and prepares for sleep, the cerebellum will gradually ease its vigilant monitoring of movement, allowing your muscles to sink deeper into relaxation. The application of pharmacological treatments to disorders of dopamine and serotonin signaling has revolutionized mental health care and provided powerful tools for alleviating the suffering of millions of people struggling with psychiatric and neurological conditions. Dopamine agonists, medications that mimic dopamine's action, are used to treat Parkinson's disease, helping to restore movement capacity in people whose dopamine-producing neurons have degenerated. Stimulant medications that increase dopamine and norepinephrine levels are used to treat ADHD, helping people with attention difficulties to focus and organize their thoughts. Antipsychotic medications that block dopamine receptors are used to treat schizophrenia, reducing the positive symptoms of psychosis, though they can carry significant side effects. For serotonin-related disorders, SSRIs are the first-line treatment for depression and anxiety disorders and have helped countless people recover a sense of hope and agency. These medications work by increasing the amount of serotonin available in synapses, allowing serotonergic neurons to have greater influence on postsynaptic targets throughout the brain. Other medications affect the synthesis breakdown or reuptake of both dopamine and serotonin, reflecting the complex interconnections between these systems. The development of these medications represents one of the great achievements of neuroscience and pharmacology, demonstrating how understanding brain chemistry can translate into real help for suffering people. As you rest tonight, knowing that your brain's chemistry is being naturally regulated by sleep, you can appreciate how the same chemical systems that are targets for medications are also regulated by your own brain's internal wisdom. Your brain is undergoing constant change, even as you rest tonight, and these changes become particularly pronounced as you move through the different decades of your life. The aging brain shows selective alterations in its structure and function, with some capabilities declining while others remain remarkably preserved or even improve. Older adults often experience a gradual decline in processing speed, the ability to rapidly perceive and respond to information, particularly when the task is complex or involves divided attention. Yet paradoxically, some cognitive abilities like vocabulary, general knowledge, and accumulated wisdom actually improve or remain stable well into old age, suggesting that aging brings both losses and gains. The prefrontal cortex, which governs higher-order executive functions, shows particularly notable changes, with dendrite density decreasing and certain types of connections becoming less stable. The decline in working memory, the ability to hold information in mind temporarily becomes more pronounced with aging, particularly when the working memory load is heavy or the task involves manipulation of information. Older adults can still learn new information and form new memories, though the process may require more repetitions and more time compared to younger adults. It reminds us that aging brings changes, but not simple, monolithic decline. Long-term procedural memories, the skills and habits you have practiced for years, tend to be remarkably preserved, even as faster, speed-dependent cognitive abilities decline. As you sleep tonight, you can take comfort knowing that rest itself is one of the most powerful interventions available for supporting brain health across the lifespan and maintaining cognitive vitality. Dopamine receptors come in several distinct subtypes labeled D1 through D5, and these different receptor types are distributed throughout your brain in patterns that create functional specialization. D1 and D5 receptors are classified as excitatory or stimulating, receptors that tend to increase neuronal firing when activated, while D2, D3, and D4 are inhibitory receptors that tend to decrease neuronal firing. Different brain regions express different combinations of dopamine receptor subtypes, allowing dopamine to have distinct effects depending on where it acts. The striatum, which is involved in movement control, expresses primarily D1 and D2 receptors, and the balance between these two receptor types appears crucial for smooth, coordinated movement. The prefrontal cortex, which governs decision-making and planning, is particularly rich in D1 receptors, suggesting that dopamine's action here is mainly stimulatory, promoting active thinking and engagement. The nucleus accumbens, involved in reward processing and motivation, contains different receptor distributions than the striatum, allowing dopamine to create motivational and pleasurable effects distinct from its role in movement. Medications that selectively target different dopamine receptor subtypes can have distinct effects, some promoting movement, while others affect mood or attention. This receptor diversity suggests that dopamine's effects on your brain and behavior are far more nuanced and specific than the popular conception of dopamine as simply a pleasure chemical. As your brain settles tonight, dopamine continues its selective conversation with different brain regions mediated through these diverse receptor subtypes. Serotonin acts through a similarly complex family of receptor subtypes with at least 7 distinct serotonin receptor types labeled 5-HT1 through 5-HT7 distributed across your brain in distinct patterns. Different receptor subtypes mediate different effects of serotonin, so that serotonin released in one brain region might produce effects quite different from serotonin released in another region due to differences in which receptor subtypes are present. The dorsal Rafe nucleus, which produces much of the brain's serotonin, sends projections throughout the brain to regions as diverse as the amygdala, hippocampus, prefrontal cortex, and hypothalamus. Serotonin acting on different receptors in the amygdala might reduce anxiety, while serotonin acting on receptors in the hypothalamus might regulate appetite and circadian rhythms. SSRIs increase the amount of serotonin available in synapses, but because serotonin receptors are distributed throughout the brain, these medications have widespread effects on mood, cognition, sleep, and many other functions. Understanding serotonin receptor diversity helps explain why serotonergic medications have both beneficial effects and sometimes unwanted side effects as the medications increase serotonin throughout the brain, affecting many different systems. The complexity of serotonin systems suggests that mental health and well-being depend on intricate balances of this neurotransmitter across multiple brain regions, and that restoring health sometimes requires more subtle interventions than simply increasing overall serotonin levels. As you approach sleep, serotonin continues its multi-system conversation throughout your brain, preparing your mind for the transition to rest. Within the temporal lobes of your brain sits a remarkable structure called the hippocampus that serves as the gateway through which all new declarative memories must pass before they can be stored permanently. The hippocampus is named for its vague resemblance to a seahorse, a shape that becomes apparent only when the structure is viewed from certain angles, revealing the brain's whimsical nomenclature. This structure contains subregions with different specialized functions, the dentate gyrus and CA3 and CA1 regions, each playing distinct roles in memory formation and retrieval. The hippocampus appears to function as a kind of temporary buffer where new information is initially bound together before being gradually transferred to the cortex for long-term storage in a process called systems consolidation. Damage to the hippocampus can result in anterograde amnesia, the inability to form new memories, while older memories stored in the cortex remain relatively intact. The hippocampus also plays a crucial role in memory retrieval, serving as a kind of index that helps retrieve stored information from across the cortex during the recall process. Remarkably, the hippocampus is one of only a few brain regions where neurogenesis continues into adulthood and new neurons born in the dentate gyrus integrate into existing circuits and contribute to memory formation. The hippocampus also appears involved in spatial memory encoding, where you are in space and the routes you have taken, suggesting that many of your memories include spatial context. This tells us that memory is not purely abstract, but rather deeply rooted in spatial and embodied experience. As you sleep tonight, your hippocampus will be working particularly hard, consolidating the day's experiences into memories that will either fade or persist depending on their significance and the quality of your sleep. Stage 3. Sleep, also called deep sleep or slow wave sleep, represents a state of such profound brain reorganization that some neuroscientists describe it as the brain's most important restorative state. During deep sleep, your brain exhibits a dramatically different electrical signature with massive synchronized delta waves indicating that large populations of neurons are firing together in coordinated patterns. This synchronized activity is strikingly different from waking consciousness and even from other sleep stages, where neurons tend to fire in more desynchronized patterns, reflecting the brain's simultaneous processing of multiple streams of information. Your consciousness recedes almost completely during deep sleep, and you can experience very little sensory awareness of the external environment, being very difficult to awaken. Growth hormone secretion peaks during deep sleep, and this hormone drives physical restoration, including muscle, protein synthesis, bone strengthening, and immune system fortification. The glymphatic system, which clears metabolic waste from your brain, operates most efficiently during deep sleep, with cerebrospinal fluid washing through neural tissue at rates far exceeding those during waking. Memory consolidation of declarative memories appears to occur during deep sleep through mechanisms involving the replay of waking experiences and their gradual transfer from hippocampus to cortex. The restorative effects of deep sleep are so profound that sleep deprivation, selectively reducing deep sleep, leads to cognitive impairment, mood disturbance, and increased susceptibility to illness. As you drift deeper into sleep tonight, your brain will gradually transition into the profound synchronization of deep sleep when restoration and repair become your brain's primary work. The region CA3 of the hippocampus contains what neuroscientists call an auto-association network, a neural circuit architecture that allows the hippocampus to perform a remarkable feat called pattern completion. Pattern completion refers to the ability to retrieve a complete memory from just a partial cue or fragment, much like how hearing the opening notes of a song allows you to imagine the entire melody. The CA3 autoassociative network accomplishes this through a circuit architecture where neurons are densely interconnected such that activation of one set of neurons tends to activate related sets of neurons. When you experience something for the first time, the CA3 network is rapidly modified through Hebbian learning, such that neurons that fire together during the experience become more tightly connected. Later, when you encounter a partial reminder of that experience, the CA3 network can complete the pattern and retrieve the full experience from just the fragment. This mechanism appears to be one reason why smells can suddenly flood you with vivid memories, because olfactory information goes directly to the hippocampus, bypassing some of the filtering of other sensory systems. The autoassociative properties of CA3 suggest that memory retrieval is not simply a matter of searching a filing cabinet, but rather involves reconstruction and pattern completion. This tells us that memory is dynamic and malleable rather than static and fixed, always being reconstructed anew each time you recall it. As you sleep tonight, your hippocampus will be replaying the day's experiences and gradually consolidating them into memories through the mediation of these remarkable pattern completion mechanisms. During REM sleep, your heart rate and blood pressure rise substantially, creating a state of cardiovascular activation that resembles in some ways the activation that occurs during intense waking exercise. Your body temperature regulation becomes impaired during REM sleep, with your skin temperature dropping to its minimum values of the night, while your core body temperature becomes somewhat elevated and harder to regulate. These temperature changes and cardiovascular changes mean that your body during REM sleep is in a state that would be concerning if it occurred during waking, but appears to be essential for the dream-based processing that occurs during this stage. Your respiratory rate also becomes irregular during REM sleep sometimes, with periods of rapid breathing followed by pauses, a pattern quite different from the regular breathing of deep sleep. Researchers believe that these physiological changes during REM sleep may reflect the intense neural activity occurring as your brain processes and consolidates emotional and autobiographical memories. The cardiovascular and respiratory changes of REM sleep also suggest that this stage involves significant metabolic activity and energy expenditure, despite the paralysis of your muscles. This physiological activation during REM sleep reminds us that sleep stages are not all equally restful, with REM sleep involving intense internal processing rather than simple physical restoration. The body's tolerance of these unusual physiological states during REM sleep suggests deep evolutionary roots to this sleep stage. As you cycle through REM periods during tonight's sleep, your body will tolerate these remarkable cardiovascular and respiratory changes, allowing your brain the opportunity to perform its most intensive memory consolidation work. Selective attention is the ability to focus on particular information while ignoring other information in your environment, and this ability allows your brain to manage the overwhelming amount of sensory information constantly impinging on your senses. At any given moment, your sensory systems receive far more information than your brain can process, so attention acts as a filter, determining which information enters consciousness and which information is filtered out and remains unprocessed. Novelty, emotional charge, and personal relevance are factors that capture attention, automatically drawing your focus even without conscious intention. Information that captures attention through one of these mechanisms has dramatically enhanced chances of being encoded into long-term memory compared to information that receives no attention. The prefrontal cortex appears involved in directing attention voluntarily, allowing you to choose to focus on particular information despite its lack of emotional salience or novelty. Divided attention, the ability to split focus between two simultaneous tasks, becomes more challenging with age and cognitive load as your prefrontal cortex becomes less able to coordinate the allocation of attentional resources. The relationship between attention and memory is reciprocal, with attention being necessary for memory formation and memory retrieval drawing on attentional resources. As your brain prepares for sleep tonight, the systems governing attention will gradually power down, allowing your conscious connection to the external world to fade. This release of attentional focus marks one of the first steps toward the inward turning of consciousness that characterizes sleep. The prefrontal cortex is the most evolutionarily recent addition to the primate brain and appears to have undergone particularly extensive expansion during human evolution. This region governs higher-order executive functions like planning attention, shifting decision-making impulse control, and abstract reasoning, all distinctly human cognitive abilities. The prefrontal cortex maintains working memory, the ability to hold information in mind, temporarily allowing you to manipulate information and solve problems using this held information. This region also coordinates and regulates other brain systems, determining which information is promoted into consciousness and how different brain regions communicate to produce coherent thought and action. The prefrontal cortex is one of the last brain regions to mature, not reaching full development until approximately age 25, explaining why judgment and impulse control continue improving through late adolescence and into early adulthood. Damage to the prefrontal cortex can result in profound changes in personality decision-making and impulse control, as famously demonstrated by the case of Phineas Gage, whose personality changed dramatically after an iron tamping rod penetrated his frontal lobe. The prefrontal cortex develops through continued experience and learning, particularly through challenging experiences that require adaptation and novel problem-solving. The prefrontal cortex also shows relatively pronounced changes with aging, with some decline in function, but also the possibility of compensation through accumulated knowledge and expertise. As you drift towards sleep tonight, the prefrontal cortex will gradually power down, releasing its executive control and allowing your brain to enter the more primitive and emotion-driven mode of consciousness characteristic of dreams. The neurons that produce the neurotransmitter acetylcholine flood the brain with this chemical during REM sleep, creating a neurochemical environment quite distinct from that of waking consciousness. Acetylcholine is released from neurons in the brainstem, particularly from the pedunculopontine, tegmentum, and laterodorsal tegmental areas that project diffusely throughout the cortex. This widespread acetylcholine release during REM sleep appears to create the conditions for vivid hallucinations and dreams as the brain generates coherent experiences without external sensory input. Acetylcholine also appears involved in attention and learning during waking, so its role during REM sleep in facilitating dream generation seems almost like a repurposing of systems that normally serve attention and learning. Interestingly, acetylcholine levels are low during deep sleep, when delta waves dominate and rise again during REM sleep, perhaps explaining the profound differences in the nature of consciousness between these stages. The balance of acetylcholine with other neurotransmitters determines your state of consciousness, with waking consciousness characterized by specific ratios of acetylcholine with serotonin and norepinephrine. The interplay between acetylcholine and other neurotransmitters determines whether you experience the external world in waking consciousness or the internal world of dreams during REM sleep. As your brain cycles through REM periods tonight, acetylcholine will wash across your cortex, enabling the vivid dream experiences that will accompany these most emotionally charged sleep stages. As you settle deeper into profound relaxation, now feel the weight of your body sinking gently into the surface beneath you, and know that everything within your brain is beginning to shift toward an inward-focused state of contemplation. The dopamine system and the serotonin system work together in intricate ways that scientists are still working to fully understand, with each system influencing the other through shared pathways and overlapping receptor distributions. When dopamine levels are low, people often experience not just reduced pleasure, but also reduced motivation and a sense of flatness, as if all experiences are muted and distant. Yet dopamine does not work in isolation, but rather in concert with serotonin, such that depression often involves dysregulation of both systems, and medications that affect one often have effects on the other. The interplay between dopamine and serotonin systems determines much of your emotional tone, whether you feel engaged, motivated, and capable, or withdrawn, unmotivated, and hopeless. Serotonin appears to modulate the impact of dopamine, such that low serotonin can amplify the effects of dopamine dysregulation and worsen mood disturbances. The balance between these two crucial systems appears optimized during restful states, allowing both to maintain their equilibrium. As you relax now, your brain is naturally increasing serotonin and maintaining dopamine balance, promoting the calm, focused attention that makes sleep possible. This neurotransmitter balance represents one of your brain's most fundamental achievements, the capacity to modulate your own emotional and motivational state through chemical self-regulation. The quiet comfort you feel as you approach sleep tonight is, in part, a reflection of these two systems working in beautiful synchronization. The consciousness that you experience moment by moment is not a simple unified stream, but rather a complex construction that your brain actively assembles from multiple parallel streams of information. The thalamus acts as a central hub, receiving information from your senses, coordinating that information, and sending it to the cortex where conscious perception arises. Consciousness appears to arise through interactions between the thalamus and cortex, with each brain region constantly communicating and comparing expected information with actual sensory input. When something unexpected happens, your brain detects the mismatch and brings it into conscious awareness, particularly if the mismatch carries emotional or survival relevance. Memory and consciousness are deeply intertwined, with the hippocampus playing roles in both the formation of new memories and the sense of personal continuity that underlies conscious experience. When the thalamus fails to function properly, consciousness is disrupted, and disorders of consciousness can emerge, such as coma or other states of profound unconsciousness. The binding of information from diverse sources into a unified, conscious field requires constant communication between multiple brain regions and appears to be something your brain actively constructs rather than a passive reflection of your surroundings. This construction of consciousness happens moment by moment throughout your waking life and continues in altered form during dreams. As your mind approaches sleep tonight, you will experience a gradual dissolution of waking consciousness as the thalamic cortical systems that generate it shift into dream-focused modes of operation. In the earliest stages of sleep, called stage 1, your brain begins to transition away from the processing of external sensory information toward an inward-focused state of gradually deepening consciousness. During stage 1, sleep, which typically lasts only a few minutes, your brain shows theta wave activity that represents a rhythm intermediate between the faster waking rhythms and the slower deep sleep rhythms. Your eyes move slowly beneath your eyelids in a gentle rolling motion, quite different from both waking eye movements and the rapid jerking eye movements of REM sleep. Your muscle tone gradually decreases, though you retain enough muscle tension to remain in your current sleeping position, and you can still be easily awakened by a stimulus. The transition into stage 1 is marked by a sense of drifting consciousness, where you may experience hypnagogic imagery, fleeting dreams, or unusual sensations like falling or floating. Your brain during stage 1 is already shifting toward lower arousal, though your sensory systems remain responsive enough that loud noises or someone calling your name could pull you back into wakefulness. This stage serves as a crucial bridge between wakefulness and deeper sleep, allowing your brain to gradually shift gears rather than abruptly switching off. Stage 1 represents your brain's first steps toward the deeper restorative states that follow. As you begin your sleep tonight, you will pass through this gentle transitional stage as your consciousness softly fades, like the sun gradually disappearing over the horizon. Stage 2. Sleep, which typically occupies about 50% of your total sleep time, represents a continuation of the transition toward deeper sleep, yet with distinctive features that mark it as different from both stage 1 and deep sleep. During stage 2, your brain waves continue to slow but show brief bursts of faster activity called sleep. Spindles that occur 2 to 3 times per minute and appear important for memory consolidation. These sleep spindles represent episodes of localized neural synchronization where clusters of neurons fire in coordinated bursts, perhaps working to integrate new learning into existing memory stores. Your muscles continue to relax during stage 2, moving closer to the deeper relaxation of stage 3 sleep, though you retain enough tone to maintain your sleeping position. Your body temperature continues to drop, and your heart rate continues to slow, though not yet at the minimal levels of stage 3 sleep. Stage 2 Sleep seems to represent a kind of stable intermediate state from which your brain can either return toward wakefulness or deepen further into stage 3. The sleep spindles that characterize stage 2 appear to serve multiple functions, including memory consolidation, protection against external disturbance, and the facilitation of sleep-dependent plasticity. As you progress deeper into stage 2 during your sleep tonight, the sleep spindles will continue their quiet work of integrating your day's learning into your brain's permanent knowledge base. This stage represents a kind of productive rest, where restoration has begun, though not yet reached the profound depth of stage 3. The limbic system is a collection of interconnected brain regions, including the amygdala, hippocampus, cingulate cortex, and others that work together to process emotions, drive motivation, and form memories. Both dopamine and serotonin send projections throughout the limbic system, allowing these neurotransmitters to profoundly influence your emotional experience and motivational state. The amygdala, which processes emotional significance, particularly fear and threat, receives dopamine and serotonin projections that modulate how strongly you react emotionally to events. The hippocampus receives dopamine and serotonin input that affects memory formation, particularly the consolidation of emotionally charged memories. The cingulate cortex, which links emotion, cognition, and motivation, receives projections from both dopamine and serotonin systems. The nucleus accumbens in the ventral striatum receives particularly dense dopamine input and shows especially strong responses to rewarding stimuli and pleasant experiences. The interplay between dopamine and serotonin in limbic regions appears to determine your overall emotional tone, whether you feel capable and motivated or overwhelmed and hopeless. Dysregulation of these systems in limbic regions is implicated in depression, anxiety, PTSD, and other emotional disorders. As you relax now, your limbic system is gradually shifting into a quieter mode as dopamine and serotonin levels adjust towards sleep-conducive patterns. This emotional quieting is essential for the transition into sleep and represents your brain's wisdom in shifting from emotional engagement to restful receptivity. Dendritic spines, the small protrusions where neurons receive synaptic input, show remarkable plasticity, changing in size, number, and shape in response to experienced learning and neural activity. When you learn something new, or when you practice a skill, the dendritic spines in brain regions related to that learning undergo rapid changes, becoming larger, more stable, or more numerous. These changes in dendritic spine morphology appear to represent the physical substrate of learning, with new spines being formed as new connections are established and existing spines being strengthened as important connections are reinforced. There are different types of spines, with thin, immature-appearing spines appearing more flexible and subject to rapid change, while mushroom-shaped, mature spines appearing more stable and structurally important. The process of learning involves a dynamic conversation between the growth of new spines and the pruning of unused spines as your brain simultaneously creates new capacity and eliminates unnecessary connections. During sleep, this process of spine remodeling continues with certain spines being eliminated, while others are stabilized, as if your brain is reviewing the day's learning and deciding which new connections are worth keeping. With aging, there is a tendency toward loss of thin spines, while mushroom spines remain more preserved, suggesting that while new learning becomes somewhat harder with age, well-established memories and practice skills remain more stable. As you sleep tonight, your brain will engage in this quiet remodeling of dendritic spines, consolidating the learning from your day into physical changes in your brain's structure. The hypothalamus is a small but remarkably influential brain region that receives serotonin projections and uses that input to regulate crucial functions including appetite, body temperature, circadian rhythms, and sexual motivation. The hypothalamus contains neurons that detect your body's current state of hunger, satiety, body temperature, and time of day, and use that information to trigger appropriate behaviors and hormonal responses. When serotonin levels are appropriate, the hypothalamus can effectively regulate appetite, preventing both excessive hunger and excessive overeating. Serotonin projections to the hypothalamus also help regulate circadian rhythms, your body's internal clock that determines when you feel sleepy and when you feel alert. The hypothalamus is particularly responsive to light, with light exposure suppressing serotonin levels and darkness allowing serotonin levels to rise, promoting sleepiness. Body temperature regulation is also influenced by serotonin acting in the hypothalamus, and dysfunction of serotonin signaling can result in temperature regulation problems. The hypothalamus also regulates the release of many hormones through its connections with the pituitary gland, and these hormonal systems are modulated by serotonin. As darkness falls and you approach sleep, the hypothalamus is receiving the signal from reduced light exposure, and serotonin levels are rising appropriately, triggering the cascade of changes that prepare your body for rest. This ancient brain region represents one of your brain's core regulatory mechanisms, ensuring that your body's fundamental needs are met. Memories of skills and habits, called procedural memories, are stored somewhat differently from memories of facts and events called declarative memories, and aging affects these two types of memories quite differently. Procedural memories of how to ride a bike or play a musical instrument or perform any learned skill remain remarkably preserved even in very old age, suggesting that the neural systems supporting procedural learning are quite robust. Declarative memories of where you went or what happened to you show more age-related decline, particularly when retrieval requires active search through your memory stores. This differential preservation of procedural versus declarative memory in aging suggests that different neural systems support these different memory types, with the procedural memory systems showing greater resilience. The striatum, which is particularly important for procedural learning, tends to show more preservation of function with aging compared to the hippocampus, which is crucial for declarative memory. This pattern suggests that you can remain capable of learning and perfecting new skills throughout your life, even as your ability to remember facts or events may gradually slow. This tells us something hopeful about aging, that while some aspects of memory become more challenging, others remain remarkably intact. As you rest tonight, your brain will be consolidating both procedural memories from any skills you practice today and declarative memories of events you experienced. Consciousness and episodic memory appear to arise through overlapping neural mechanisms, with both depending on interactions between the hippocampus and cortex, and both mediated by similar molecular processes. Episodic memory. The memory of events that you can consciously recall appears to depend on the same binding processes that generate unified conscious experience in the moment. When you consciously experience an event, your brain is binding together visual information, auditory information, emotional information, and spatial context into a unified experience, much as when you consciously recall that event, you are reconstructing all these elements from partial cues. The hippocampus appears central to both conscious experience of the moment and the later conscious recollection of that experience, suggesting that consciousness and memory are fundamentally linked. The medial temporal lobes, including the hippocampus, appear crucial for conscious awareness, with damage to this region resulting in unconsciousness or profound memory impairment. The interplay between consciousness and memory suggests that both arise through similar neural mechanisms involving the binding and integration of information from multiple sources. As you drift toward sleep, consciousness will gradually recede as the thalamic cortical systems that support waking consciousness power down. Yet your hippocampus will remain active, continuing its work of consolidating your day's conscious experiences into memories. The rhythm of sleep progresses through stages in a cycle that typically takes about 90 to 120 minutes from the beginning of light sleep to the end of a REM period. You experience multiple complete sleep cycles each night, with 4 to 6 full cycles occurring over an 8-hour sleep period. The later sleep cycles in the night tend to have longer REM periods and shorter deep sleep periods, suggesting that your brain prioritizes different types of sleep as the night progresses. The first cycle of the night tends to be dominated by deep sleep stages, as if your brain is most hungry for physical restoration early in the night. By the end of the night, the cycles are dominated by REM sleep, suggesting that your brain prioritizes emotional and cognitive processing as the night wears on. This cycling through different sleep stages occurs automatically without your conscious involvement and represents your brain's sophisticated scheduling of different restorative processes. The quality of sleep appears affected by whether you allow these complete cycles to unfold, as cutting sleep short eliminates the final REM periods that occur only late in the night. Understanding this cycling pattern can deepen your appreciation for the importance of getting sufficient total sleep duration. As you begin your sleep tonight, you will enter into this ancient rhythm of cycling sleep stages, allowing your brain to orchestrate its own complex dance of restoration and consolidation. As sleep continues through the night and you cycle through repeated stages of sleep, your brain remains partially active, coordinating the transition between stages and continuing the ongoing work of processing, consolidating, and integrating information. The brainstem, which sits at the very base of your brain, plays a crucial role in regulating sleep-wake cycles, coordinating the brain regions that control different sleep stages. The brainstem contains neurons that produce serotonin in the Raphe nuclei neurons that produce dopamine in the ventral tegmental area, and neurons that produce acetylcholine in the pedunculopontine tegmentum. The coordinated activity of these different brainstem systems determines whether you are awake in light sleep, deep sleep, or REM sleep, as different combinations of these neurotransmitter systems create different brain states. During deep sleep, the arousal systems of the brainstem are relatively quiet, allowing the thalamus and cortex to engage in the synchronized delta wave activity characteristic of this stage. During REM sleep, the arousal systems are reactivated with acetylcholine flooding the cortex, while serotonin and norepinephrine are suppressed. The brainstem appears to operate with a kind of biological wisdom, knowing when to activate sleep systems and when to activate arousal systems based on your circadian rhythms and sleep pressure. As you progress through your night's sleep, your brainstem will continue its coordinated orchestration of the different sleep stages, ensuring that you move smoothly through cycles of restoration and consolidation.